Hello, it's March 30th, 2015. My name is Marc Kaufman, and I'm here to interview Dr. Jerome Dempsey for the Living History Project of the American Physiological Society. Dr. Dempsey, can you tell us a little bit about your family upbringing and how you became interested in science?
Yeah, I'm a Canadian, born and raised in London, Ontario. My interest in science really didn't start till high school. And I had a biology teacher there that was turned on by that. It was my best subject along with math, so I guess I had kind of a predilection in that area. But the interest in science, I don't think really started till partway through university. And there I was influenced by a couple of people. One man's name is Mike Uhas. He wasn't really a scientist, but he was getting his, he actually was my football coach. I played a year of football. That only lasted a year because 160-pound linebackers don't last very long, and even Canadian football. But he was great at that, and I saw what he had great enthusiasm for, he was a fitness guru, and he kind of researched that too. And that kind of got me started and interested in that. But I really wanted to be a teacher, high school teacher and a coach. And when I graduated, that's what I did. This was my first job.
How about what got you interested in going to graduate school?
Yeah. So I spent that year teaching in a place called Hamilton, Ontario, and I thoroughly enjoyed it. Coached 4 sports, taught 3 different subjects. That's what you did back then. And the Canadian government, so this is the early 60s, got very interested in sport and exercise, and they created scholarships for the first time, very early in the 60s. And to this day, I can't remember exactly how this man got my name, or I got his, but an Australian by the name of Max Howell was at the University of Alberta, and he came to the city I was in teaching to interview people, and I interviewed with him. Geez, it was very exciting what he was talking about. So I decided that 1 year was enough of teaching and that I'd go back to school, fully intending to come back and teach further. And then that 1 year I was getting a master's degree in exercise physiology at the University of Alberta. I met another man named Brian Spruill. He actually started pulmonary medicine in the western provinces of Canada, and he became very famous for that. But he had studied with Jerry Mitchell at Southwestern in Dallas, Texas, and they published a very famous paper called The Physiological Meaning of the Maximal Oxygen Uptake in the late 50s. And so I got a little connected with Spruill and went over to his lab, saw what a pulmonary lab was like. He helped me, or some of the people that worked for him, helped me with the project that I was doing. And there was no turning back after that. It got very exciting, and that's what really got me going in science. Yeah.
A topic that we've had many conversations over and is sort of near and dear to our hearts is the issue of mentorship for young students and postdocs. Yeah. Tell us a little bit about the mentors who played an important role in your career.
Oh yeah, I agree. And I was really, really lucky because after a short while at Wisconsin, I did not go down there because of him. I met a man by the name of John Rankin, and he was a pulmonary physician from Glasgow, Scotland, and he had immigrated to the U.S. and was on the faculty at the University of Wisconsin. And he started this little lab to look at exercise in the lung. I remember the cardiologists had this huge suite of rooms, and they gave us their lunchroom to start this lab in. And there was another graduate student, 2 other graduate students, and me. And we were interested in diffusion in the lung and how exercise affected it and how training affected it. And He was really a wonderful man, a great listener, extremely bright. I tended early in my career to shoot my mouth off a little more than I should have. Probably continued that.
That comes as quite a surprise.
Yeah, it's very surprised to you, I'm sure. and he was very protective of me. When I'd say to him, "Oh God, I've kind of done these," it's okay, good spirit, you know. I mean, he kept encouraging me to question and to be skeptical, and how that would help your own research is not just criticizing other people, but how it would help your own. And I think he was right in the long run. But so I mean, that was terrific. And then I wanted to go back to Canada, but I think if it had been anybody else, I would have, because I really wanted to go home. But he was such an attraction of that place, and he unfortunately died in his late 50s. But my first 15 years on the faculty, or 12 or 14 years on the faculty of Wisconsin, he was my boss as well as my mentor. And he was so supportive in so many ways. Even though I can remember he had a program project grant. And I had one of the projects on interstitial lung disease. He was an environmental lung disease specialist. And I was working on the project, everything was going okay, but I really wasn't that fired up about it. And I got interested in the control of breathing. And got my first graduate student, Bert Forrester from Milwaukee, or from rural Wisconsin. And that's what I really wanted to study. And I think it was a couple of weeks or a couple of months before the big site visit that was coming, and I said, "Dr. Rankin, I really don't want to do this anymore. This is the direction I want to go." And he gave me his blessing on that.
Wow.
You can imagine
Wow.
somebody that selfless that would do that. It's very selfish of me, but he saw this, and so that was an example of what he was like. Yeah, very, very important to me. There were other mentors too, and I can go way back to I was raised by a single mom whose work ethic was unbelievable. I had a terrific older brother as a model, but I think certainly professionally, Rankin was it, and a great legacy that he left. Yeah.
I just can't help but emphasize how important I think mentorship is, especially the one-on-one relationship, and where people are role models for both behavior, attitudes, and values. And I think that is so important for the training of young scientists today.
And I think, to me anyway, I'm not even sure I realized this for about 20 years, but it really became my main job and my main worry. I always worried, are we providing worthwhile enough experience for these people? And as far as our lab that became known as the Rankin Laboratory of Pulmonary Medicine, which existed almost 50 years, including my time there, when we're pretty well done now. But our most important legacy is the 68 pre- and postdoctorate fellows that we trained in that lab. And so I agree with you. I think that at least to me, that became my focus, these people. Certainly you do the research, but and you do the research and you search for what's true and all that. And that's important. But the mentoring of these people is absolutely essential. And I think it certainly for me also gives me the most pleasure and the most kick and the most excitement. And now seeing these young people now, I just got back from Japan. There were 3 of our former Rankin trainees there. And seeing how they've come along, including their struggles, and what, that's very satisfying. Yeah.
I can imagine.
I agree.
Yeah. Let's talk a little bit about your research, and I was hoping to do it in a chronological order.
Sure.
And the first thing that comes to my mind is ventilatory acclimatization. And I always, I guess, not to be flippant, but I sort of see you as David versus Goliath, and David won. And I was so impressed with that. Can you tell us just a little bit about it?
Just a little bit, because I'm not sure about who won or anything, but
It took a lot of courage to do what you did.
Well,
I was very impressed by it.
in the in the late 60s, so I'm just finishing graduate school, really. And my first graduate student was, as I said before, was Bert Forrester. And it's an interesting story, actually. We got interested more in the control of breathing rather than gas exchange in the lung. And although I've always had an interest in all aspects of respiratory physiology and researched them, because I think they're all so interconnected. And Rankin really pushed that, too. So we said, well, the only way to research this is go to the mountains. We knew nothing about this. We'd done exposing ourselves and knocking ourselves out with hypoxia that was too severe. This was the days before internal review boards were very serious. and we decided we need to do that. So I went to Dr. Rankin and I said, you know anybody in Colorado? He knew somebody, phoned him, he wasn't the one, but he gave us a name, Bob Grover, Robert Grover, a very famous guy in the pulmonary circulation and in hypoxia. Most wonderful man. And this would never happen today, I can't believe it, but we contacted him. He met us at an FACIP meeting, or experimental biology meeting, about a month later. And he said, these are the words he used, he said, I've got a grant from the Department of Defense to look at hypoxia, and we've answered the question, but there's another year left on it. I could support the work you wanted to do. Imagine.
Wow.
What a wonderful thing to do. And we did. We did. And we did meet with the Department of Defense people at that meeting for about 20 minutes for what that was worth. But it was Dr. Grover who did this. So we went out, starting in 1967, with hypoxia. And there was a prevailing theory at the time that, we knew a fair bit about the carotid chemoreceptors and their role in hypoxic sensing. Okay. But why did ventilation keep increasing over time at hypoxia? That was a mystery. And what about these newly found receptors called central chemoreceptors or medullary chemoreceptors that were responsive to acid? How did they fit in? And the group in San Francisco, led by Dr. Severinghaus, had this wonderful concept. And we all bought it. And it was a way of explaining not just acclimatization to hypoxia, but many other clinical conditions, the control of breathing in most acid-base conditions. And he and Bob Mitchell came up with this theory, and it really changed the field. I remember teaching it to medical students when I started to teach. We bought it, but it was based on a minimal amount of data and a lot of assumptions. And we knew that, but it still made such a beautiful story about how the peripheral and central chemoreceptors interacted. And the key was that the hydrogen ion in the brain extracellular fluid was regulated quite differently than it was in the blood. That was their presumption, their claim. And it was, in order to do that, was probably active transport of chlorine bicarbonate across the brain. Another great story. But if you look carefully at the literature, stuff that was not even on that topic, there were holes in it. I remember in anesthetized animal studies from Boston, Kazemi's work, looking at something and say, well, the brain extracellular fluid doesn't look to be regulated any better than the blood. So we put that to the test. And we started off by doing it in ourselves, which means you have to take spinal fluid samples. And we did that. We found out that they were at 3,000 meters altitude, they were identically regulated. So in other words, this central chemoreceptors couldn't have been playing much of a role because they were becoming alkaline as we were breathing more, just exactly the opposite way. But people weren't buying what we were saying, and so we would get more subjects. And then we went to a pony model. This is where Jerry Biscard came in. So this trio of us, of Forster and me and Biscard, worked for 10 years on this. 3 different species, a dozen different studies, different ways of looking at it. Still, in every experimental biology meeting, the voices would get loud, and the controversy was great, very exciting. And then a guy who'd really been helping us a lot, a Czech refugee by the name of Vladimir Fencel, who died about 13 years ago now. He knew acid-base balance better than anybody that I ever knew. And he wrote beautiful methods sections in his papers. He worked with John Pappenheimer at Harvard. And I would write him about it, talk to him about it. And he finally decided to repeat one of our studies. And he found what we found, not the other presumption. And then somebody in San Francisco was actually asked to kind of act as a referee in this, Ralph Kellogg, the late Ralph Kellogg, and he wrote an article summarizing all the available data. Then there was a meeting in India, chaired by Paintal, famous Indian physiologist. A lot of famous people got involved in this. And then Tom Hornbein, the doctor who climbed Everest, was the chair of the session that went on into the evening. And I think that was kind of the point where people said, yep, it looks like we've got to look for something else. And that's where people came in and said, well, can the carotid body actually change? And then Jerry Biscard was the main one moving there and showed that indeed it did within hours, actually. So that's where I think that there's still some questions to answer, actually, about the field. But anyway, that was my introduction to heavy-duty research in respiratory physiology.
You mentioned I guess these were American Physiological Society meetings in those days, rather than EB, but the forerunner. There were a lot of battles and arguments at these meetings. Did you also have trouble getting papers published?
Yeah, at the very beginning.
And for a young person, this could be very discouraging, but yet you pushed through and you turned out to be right. I find that remarkable.
Yeah. Well, thanks, I think. But I guess, is there a lesson to be learned from that? Yeah. I remember the first paper was published in Journal of Clinical Investigation, our first human paper. And it ended up having about 6 reviewers, and it went in 3 different times, and blah, blah, blah. After that, it got a little bit easier, but it's still, to this day, I read a review a month ago, and somebody was saying, and here's the way it's done, and in the textbooks, that the older idea was held onto for a long time because it was so straightforward and such a beautiful explanation. But yeah, it took some time, but I don't know whether that's unique with us. I think that people have to persist and certainly be skeptical of your own findings. Well, you and I were at a meeting this morning in which some of our own data, this is kind of interesting now, because now I'm not the 27-year-old, 30-year-old anymore. I'm a little older than that now. And now we're questioned by younger people. So it comes full circle. And so that's a test of how obstinate am I, or how single-minded am I? And I found it challenging. But trying to remember from what I went through and what Bert Forster and I went through, that was a good lesson. Yeah.
But I think, as you say, controversy results in progress.
Oh, yeah.
And I think, we need a thick skin. I think that's really important. And a small ego. I think those are all very important.
Yeah, and self-criticism.
Yes, yes.
I think, don't you, I know in our lab meetings over the years with these wonderful trainees, I always felt, I don't know whether I was consciously thinking of this, but it just evolved in our lab meetings. And it came from those controversial years in the 70s. I mean, we would practice that, okay, when we're giving our paper at the next meeting, we'd get somebody to act as 2 of the San Francisco guys and just throw these hard questions at us and raise voices and all that. And in our labs meetings, I thought the most important thing that the trainees had to learn was to relish the fact that they were getting difficult questions, to relish that and to make them prepare better. So, yeah, there are a lot of lessons from that.
I think that's very good.
Yeah.
Let's talk a little bit about sleep apnea, because that was another important issue. Can you tell us a little how that got started?
Yeah. Well, isn't that an interesting thing? I mean, we've known about apnea since the 19th century. And in Dickens, in 1837, he described this hypersomnolent, overweight young man, and then so these cases would come out like this. A couple more from Britain. Shane Stokes' respiration was discussed in Ireland in the 19th century. But nobody ever thought it amounted to anything. Nothing was ever done. Well, why are these symptoms like that? Why are people breathing like that? Why only in sleep? And then in the 50s, the so-called Pickwickian syndrome was described of obesity and hypersomnolence, but it wasn't linked to sleep again. We had a case that I won't go into, but when I was a graduate student, and we wondered why this person was breathing the way he was. Perfectly healthy person. I wrote a case study on it and published it years ago. And I thought, I wonder if it's got something to do with him consciously controlling his breathing. And so I got in touch with a neurologist at our place who did EEGs. There were no sleep lab. There was no sleep medicine then. And I said, let's study this guy during sleep. So he came over to our lab. I remember we put a big foam cushion down on top of our treadmill for God's sake in our lab, and put these on him, and glued a mask to his face. Glued. I hope the glue we used was appropriate. Anyway, he slept all night. We measured his breathing all night, and all these quirks that he'd had responding to CO2 and stuff went away. And I'll never forget how beautiful his breathing pattern was during the night. So this is 1964.
Wow.
Now, why wouldn't I have picked up on that and thought, this is something that should be studied? And, but I didn't. A paper was published out of Scandinavia at the time, and I thought, oh, this guy's answered all the questions about sleep. And of course it wasn't. It was purely a descriptive study. It was a heck of a start. So then I didn't do anything about it until 1979. And by then, even though it hadn't been fully described or certainly researched, some people at Stanford, a guy named Remmers in Texas, had kind of given us a pretty good idea that the upper airway was involved in this. Another thing about mentorship, the people in our lab weren't just PhDs. We had several MDs that trained with us. And I had a lot of really good clinical colleagues, but the one I had that taught me the most is one that I trained, is Jim Scatrid. He fortunately died of lung cancer about 10 years ago now. And I worked with him for 30 years. And he and I studied our first sleep apnea patient in 1979. Fascinating. The fact that a human could go to sleep and you could study him in a state in which really the suprapontine areas of the control system were kind of out of the picture. Wow. No anesthesia required, sleep. The only thing that was a problem is that the upper airway did get in the way. I mean, it's not well controlled in a human during sleep. So Jim and I used that and studied sleep for a couple of years, and then we with the help of people like Remmers, who I used to bounce ideas off of all the time, we found that sleep, what we will call it unmasked, a highly sensitive CO2-induced apneic threshold. And that was that led one thing to another, that the nervous control system for the control of breathing, besides the control of the upper airway, probably had a lot to do with sleep apnea. It's not just an anatomical problem. And so that really started us going on that. And we use sleep a lot in testing mechanical effects on the control of breathing, because you can ventilate people, normal people, sick people during sleep, beautifully, through the nose. I remember back those days we didn't know that. And in order to test for this apneic threshold, I had a clinical friend of Jim's intubate me.
Through the nose?
Through the nose.
Wow.
That was terrible.
No anesthesia? No local anesthesia?
No, until they sleep.
Yeah. Wow.
The problem was that we inflated the balloon too much and I couldn't sleep. Sometimes sleep I just swallow. It was a complete bust, the experiment. But we got the idea. Then one thing led to another. I thought, well, what are we doing? Why don't we try ventilating through the nose? And blah, blah, blah. So it went from there. So sleep apnea, and I still, to this day, that's the little bit of research I do these days is on sleep apnea. And it was wonderful for an integrative physiologist to study. And the collaboration with all those clinical people over the years. And then we got together with epidemiologists, because I was in a department of preventive medicine that Dr. Rankin chaired. And physiology was like a duck out of water in there. But the one contribution we did make was that we, in 1987, there were finally, and I said after a series of meetings that, look, this sleep apnea thing looks like it might be important. Although physicians didn't know about it, nobody was diagnosing it, and they decided to establish 3 centers across the U.S. to study this. Well, Jim Scatroot and I were just on our own study in the sleep stuff, physiologists, and I didn't figure we could compete for that grant, that center like that. So I talked to the epidemiologists at our place, Terry Young and biostatistician Mari Palta, really good at what they did. And they weren't physiologists, they didn't know anything about sleep apnea, no idea. So we introduced them to that, and they set up what's now become a very famous study called the Wisconsin Sleep Cohort. And that still goes today, of a little over 2,000 randomly selected state workers who come in every 4 years for a full sleep study overnight. And you can imagine the data that's accumulated. Now there's mortality data on that.
Wow.
So this has been a very powerful study. And I'm kind of ashamed to admit that the most cited study I have is an epidemiologic study, as a third author in the New England Journal of Medicine from 1993. But it was important in putting sleep apnea, I think, kind of on the clinical map, that it was a significant portion of the general population, not a clinical population, but the general population had sleep apnea. And of course, the prevalence has doubled since then because of the obesity epidemic. That's a major risk factor for it. So that's the kind of, I guess over the years, that the control of breathing and then sleep apnea, and exercise has been a major emphasis in our lab. So we looked at cardiorespiratory interactions, and most recently the role of the things you study, the 3,4 afferents, and thank you for all your input on that. And that's been another fascinating thing to get involved with.
I apologize for embarrassing you, but I really would like to know, how many citations does that Wisconsin sleep study have now?
Oh yeah, it's in the 20,000 or something.
Yeah, I mean, that's amazing.
Yeah, it is amazing.
That's really wonderful.
I'm not sure everybody cites it correctly. Seriously.
Would you like to correct it now?
No, I mean, we all evangelize a little bit.
Sure.
You know, we want our disease to be important.
Yeah.
And what is, very briefly, what is important? How much sleep apnea you need for it to be important? I think we got a handle on that now. And it's a fair bit to really know that what's causing hypertension, what's causing cancer has been related, what's affecting mortality, what's affecting insulin resistance. And you need a fair bit. And so people were taking the low number, like 5 events per hour, which I to my mind, still to me is not significant, and saying that's, the prevalence of. So that's the way I think it was wrongly cited. But I think it was a very useful and important. And these epidemiologists and biostatisticians did a really good job. It's longitudinal data, random selection, all the bells and whistles.
It's not easy to do.
Yeah.
Not easy. When did, with respect to sleep apnea, when you first started studying it, I would imagine, and tell me if I'm wrong, that you really had no idea that this would contribute to hypertension as a disease.
No.
How did this get connected?
Oh,
Because this is
really
tying in respiratory physiology with cardiovascular physiology.
Yeah. Well, we certainly didn't discover that. But what the thing that goes along with the sleep apnea, Mark, is this intermittent hypoxemia. And I guess the important part of that's much worse than its consequences, especially cardiovascular consequences, are much worse than a constant hypoxia. And people think it's the resaturation phase that starts the increase in redox state, and that goes from there, inflammatory response. And a guy by the name of Rick Fletcher at the University of Louisville Medical School I'm pretty sure he was the first one that said, what about this intermittent hypoxia? What's it doing? And he got a rat model going where he reproduced that intermittent hypoxia that people get. and saw the hypertension develop. Elliot Phillipson, who's really important to the physiology of breathing and sleep, did this in a dog model where he saw over a 2-week period the hypertension develop, and it went from there. I married a cardiovascular physiologist, Barbara Morgan, and she did a beautiful series of studies in both rats and humans on sympathetic activation and how that when the intermittent hypoxia, just give it for half an hour or so, you stop it, but it continues high, this after effect that's an effect of the carotid body and then an amplification centrally of that carotid body signal. So sleep and the cardiorespiratory response just bred all this wonderful integrative physiology and pathophysiology.
Yes.
But I think it was that kind of thinking through the early 90s, really. But then there was, because if you just look at it in the patient and you see hypertension, well, you've got a person with diabetes, usually, lots of times, some type of, and overweight, and you think, it's not the sleep apnea that's caused, it's that. So you needed the experimental model. Now, the last 5 years, 2 or 3 labs in the world have actually taken healthy young people and exposed them to intermittent hypoxia for 8 hours a day and are seeing the same kind of things develop. High sympathetic nerve activity, blood pressure rising, insulin resistance going down. Yeah, it's for real and something to be concerned about. Yeah.
Shifting gears, tell us a little bit about your interest in lung mechanics, and maybe was that done in Montreal with Milik Emily in the start?
Yeah, I took 2 sabbaticals. The first one was in Sweden in 1973 and 4, and I studied airway mechanics there with a young man by the name of Björn Backe, and I learned a lot from him, and Gunnar Grimby as well. Very famous guy in exercise and respiration. And so that was my first battle. The second one was in McGill in the early 80s, and I really wanted to study the diaphragm. The people at McGill, Peter Macklem, the late Peter Macklem, had really gotten people excited about the diaphragm at the time because they showed with some really simple studies that maybe it actually fatigued even a normal person with very non-physiological type breathing against a resistance for long periods. And I got thinking, I wonder if this ever fatigues an exercise. No way that would ever happen. But that's what got me interested in that, because I was very interested in is the lung or respiratory system ever a limiting factor to exercise? Because up till 1983, we never only studied ourselves during exercise with arterial catheters and esophageal balloons and things like that. And none of us were fit enough to ever push the respiratory system enough to see this. But then, pure serendipity, we studied a bunch of endurance athletes and found that about half of them showed that they get hypoxemic during exercise. That was a real surprise to me. And that's been confirmed. It's not everybody, and we still don't know all the reasons why. I'm not going to thinking, the respiratory system isn't overbuilt for everybody. Maybe there's, and I saw that in the lung, studied that for a while, especially in women. We saw that another 10 years later. But what about now the chest wall? And learn those techniques. Blocking on the name of the other guy at McGill, it was the Argentinian, it was so important. And brought them back to our place. And then Bruce Johnson, who was a wonderful doctoral student, spent about 6 months, he and the late Kurt Soppe, another wonderful student, trying to get this technique of phrenic nerve stimulation, looking at fatigue of the diaphragm. Set up the study, fully expected to find nothing, that the diaphragm was too good, his blood flow was too good, his mitochondrial volume was too good, it's not going to fatigue, even though there'd been some indirect evidence that it might. So we set up the study to really fail, blah, blah, blah. Well, it turned out we were wrong, that if you exercised at a high enough intensity to exhaustion, the diaphragm did show fatigue. And then we went from there to wonder, does it require blood flow? Is there, my God, yes, if you unload the diaphragm, it looks like blood flow to the limb, and vascular resistance goes down, flow goes up. And why is that? Is there a reflex, like the ones you study from the locomotor muscles, or one from the diaphragm? And so, again, that we didn't start that. Earlier work at McGill indicated that. A former student of yours, Janine Hill, to my understanding, is still the only person to have studied those 3-4 receptors from the diaphragm. Boy, does that need study. I know how difficult those must be to record from. But so it started all with that, and then we tried to apply it to the human. And so isn't it funny how little things like that, one leads to another. And if I really think if I hadn't been married to a cardiovascular physiologist, that I wouldn't have gotten into that.
Wow.
Because she could measure the muscle sympathetic nerve activity. I didn't even have enough smarts to know in the first study we did we measured blood flow, big invasive technique with thermal dilution. We had balloons down the esophagus to measure everything. I didn't even think to measure arterial blood pressure. So I could calculate resistance and conductance. And it didn't even occur to me. And I remember going home that first night and showing it to Barbara. I said, look at this, my God, the blood flow changed when we unloaded and loaded the chest wall. What's going on? Oh, she said, oh, this and that, interesting. What happened to resistance? Oh, stupid. Of course, from then on. So, I mean, you get lucky, people that you associate it with contribute a lot. And the cardiology fellows that we had then, and Peter, my good friend Peter Hansen, who arranged all that, he's a cardiologist. You wouldn't do any of these studies about people like that. So, yep.
Let's talk a little bit about intrathecal injections and the exercise pressure reflex and its control and breathing, because I think those are fascinating studies. And
yeah.
I always wondered if you ever tried it on yourself.
No, I didn't.
I wouldn't either. I'd be too scared.
Right. I did a lot of...
You've done many things.
In the 70s, we did spinal taps. And a lot of them.
Yeah.
Over
and
150.
for the sleep apnea study through the nose, I'm very impressed with that.
A lot of that.
Yeah.
And, of course, that stupid thing with the intubation. Right. No, we didn't, because we needed, again, really fit young people that could do all this exercise. I was a little above the mean age by then.
But still fit.
But that idea came from a clinician, because we wanted to study Marcus Alman, wonderful postdoctoral fellow. He was interested in performance and what mechanism really made your motor cortex, your motor areas say, maybe this is enough, quit pushing so hard. I'm sorry to oversimplify the question, but that's really what. So before it had been, the studies before that have been done with this have been done with lidocaine, epidural lidocaine. And the problem with that is that it not only blocks afferents, some of them anyway, but it blocks efferents, makes the muscle weaker. So if a weaker muscle, we know from Curare studies, means that the central nervous system is going to try and compensate for that to accomplish a given workload. So your model's wrecked, in essence, with that, I think. But we use lidocaine anyway. I remember postdoc and I arguing about that. Went into study with it. There was the weaker muscle. There was all the evidence for central command. And said, okay, although it did kind of confirm the hypothesis about the feedback being important to motor output. So then the anesthesiologist, Les Proctor, who did the lidocaine studies for us, he said, why don't you guys use fentanyl? Fentanyl? Geez, that causes respiratory failure. And, oh, no. That's when you give it intravenously. That's when it gets to the brain. But he has very lipophilic. It'll stay right down there in the lumbar, really. So we had to convince the IRB. And they were wonderful. I had not had great relations with IRBs, but they actually sent somebody over to our lab for an hour, discussed this with the anesthesiologist, Marcus and I, and they let us go ahead. We did several studies with it, and it showed not only this business about the locomotor, control of locomotor output, but again, serendipity, we breathed less, and the pressure response was greatly blunted with this. It isn't a perfect model. It's only a partial block. And there's some findings that need explaining, and a lot more needs to be done. But I think it was an interesting approach. It seems to be the drug of the month around here. A lot of people are using it. You've used it in COPD patients,
yes.
heart failure patients, and we heard again today, normals again. So, yeah, that was a neat.
I think
neat
that
trip.
was a real major advance.
But again, a
I
clinician.
congratulate you on that.
Thank you.
Yes.
But again, a clinician
Yeah.
gave us that insight to use that. Yeah.
Well, our time is almost up, and I was just thinking that maybe you'd like to say a few words to young people about futures in science, or whatever you'd like to tell them. I'd
Yeah.
like to hear it.
Yeah. It's hard for young people right now
It is.
to get
It
into
is.
science.
Yes.
They see their mentors working all the time, applying for so many grants, and the percentile level is so low. And we've gone through this before. In the 80s, this happened, and that's when the enrollment at business schools peaked, and science went way down, and we might see that again. So it's difficult. But I think it's so important early on to latch on to a mentor and pick that person really carefully. Not just for convenience, because it happens to be the same school you happen to be at, and interview strongly and find out all you can about this person and talk to other people that they've trained and stuff. I think that's such an important decision to make early on. And I guess the other thing I'd say is to be skeptical and ask those hard questions right from the beginning. And if people say, oh, what are you talking? Don't let yourself get beat down. Oh, if you're wrong or whatever, certainly realize it, but hang in there and depend on your colleagues and good mentorship. And once you get into it, it's a wonderful profession to be in. And then you become a mentor yourself, and of course that's the glory days. That's when you and your trainees accomplish things together and work together. That's the real joy of it.
Thank you very much.
Thank you, Mark.
Thank you.